substance which is diffusing in or out, it basically interacts with the carrier protein, it binds with the carrier protein, and then it moves in or out along the concentration gradient. But it has a maximum, it has a limit to its rate of diffusion, which is known as Vmax. And this limit is because of the fact that the molecules, they have to bind to these carrier proteins. So obviously this factor basically limits the extent to which the process of diffusion or rate of diffusion can increase. Okay, so we have discussed that already. Now we will just see those factors which affect the rate of diffusion. So these are the various factors that can affect the rate of diffusion. So obviously the first factor is the concentration difference across a membrane. So if the concentration of a particular substance is higher on one side and lower on the other side, like you can see over here across membrane, then obviously these ions or molecules, they will move from the area of higher concentration to the area of lower concentration. So you can see, so this is pretty simple. And the example is that of alveoli. As you know, these are present in the lungs, and this is where the gaseous exchange takes place. And that gaseous exchange basically takes place because of this concentration difference. Because the air that we are inhaling, it is rich in oxygen as compared to the blood that is coming to the lungs for oxygenation. So oxygen molecules, they diffuse into the blood, and at the same time the carbon dioxide, whose concentration is higher in that blood, that basically diffuses into the alveoli. So that's how, because of this concentration difference of these gases, they diffuse from either the alveoli to the blood or from the blood to the alveoli. So next point is membrane electrical potentials. Like if there is a certain polarity that in normal cells there is a negative charge present inside the cell and positive charge present outside the cell, or if we consider this diagram in which they have shown a positive charge on the inside and negative charge on the outside. So because of this presence of this polarity, the negatively charged ions, they will move from the area of negativity to the area of positivity, because obviously the negative ions will go towards the area which has more positive charges, right? So this is the effect of electrical gradient or electrical potential on the movement of these charged ions. So negatively charged ions will go towards the area of positivity, and positively charged ions will tend to go towards the area of negativity. But I would also like to explain another point over here, that you see these particles, negatively charged ions, are moving in this area, but there comes a time when the the concentration difference is achieved that it does not allow further movement of ions across the membranes. So I will discuss that in the coming lecture. Okay, you can just forget this right now. ये मैं आपको बाद में explain करूँगी. So you just have to go to the area of positivity, and positively charged ions will go into the area of negativity. And this happens in neurons. Okay, we will study in the next chapter that how when action potential is generated, then how these differently charged ions move, in the areas where they have the opposite polarities, right? And the third factor that can affect the rate of diffusion is the pressure difference that exists across a membrane. So over here they have just shown this side, and it is basically, because of that increased pressure on this side, the molecules, they tend to move towards the opposite side where there is lesser pressure, obviously. So the pressure gradient also affects the rate of diffusion. So where does this pressure difference exist? This pressure difference exists in the capillaries, right? Pressure due to some, fluid overload in the body, that tends to cause the diffusion of fluid from the capillaries into the surrounding areas. So that is basically the effect of pressure difference on the diffusion of molecules. Okay, so there are these three factors that can affect the rate of diffusion, concentration, the pressure differences that exist across the membrane. So moving on to osmosis, and it is basically the net diffusion of water molecules across a selectively permeable membrane. Obviously, water molecules move from the area of high water concentration to lower water concentration. And I think all of you are familiar with this concept of osmotic pressure, osmolarity, and osmolarity. Only thing that 300 milliosmoles per kilogram. So just keep this value in your mind. So up till now, the different types of transport that we have studied about, the simple diffusion and the facilitated diffusion, these are, or osmosis, all these modes of transport are, passive mode. These are basically the passive transport mechanisms, because in all those types, there is no utilization of energy. There is no utilization of adenosine triphosphate. But when we use the term active transport, it is basically the type of transport in which energy is utilized. Okay, so because in this the cell membrane, by uphill movement we mean that the movement is against the concentration gradient, or it is against the electrical gradient or the pressure gradient. So, to
मैम आपकी आवाज नहीं आ रही सही से
of lines of particles against these gradients.
मैम आवाज
आपकी आवाज बहुत डिस्टॉर्ट हो रही है.
Nothing more I can do about it. चलो फिर लेक्चर खत्म कर दें.
Yes, ma'am.
आप मुझे बताइए कि मैं कैसे इसको कर सकती हूँ मसले को हल.
मैम आई थिंक वो सिर्फ आवाज चल रही थी इसलिए रोक गया था. अभी फिर ठीक है. जस्ट रिपीट द लास्ट 1-2 मिनट.
Okay. So I just repeat it once again that when we are talking about active transport, it is basically the type of transport in which there is, the movement against the concentration gradient or electrical gradient or the pressure gradient. And to cause such a movement, it requires consumption of energy, right? So all those types of movements in which energy is utilized, in which ATP is utilized, it is known as active transport. And it has two types, primary active transport and secondary active transport. So in primary active transport, energy for the uphill movement of ions or molecules, it directly comes from the breakdown of high energy phosphate compounds. Now, I hope all of you know that the adenosine triphosphate has 3 high energy phosphate bonds attached to it. And by breakdown of every phosphate bond, a high amount of energy is released. So, when these phosphate bonds, they break down to cause the uphill movement of ions. And first thing is the utilization of energy. Second thing is the presence of carrier proteins. For active transport, the presence of these carrier proteins is really, really important. And basically, the particles or ions to be transported, they attach themselves to the carrier proteins, and then they are transported in whatever direction they are supposed to be transported. And these carrier proteins, they also have that ATPase activity, that it has the ability to break down ATP into ADP. So that energy is released and the movement of ions or particles can take place against the gradient, whatever gradient it is. So the first example of active transport, or I should say the primary active transport, it is the sodium-potassium ATPase pump. So as you can see over here, this is basically the sodium-potassium pump, and it has a binding site for 3 sodium ions on the inside and 2 potassium ions on the outside. So when both these ions have bound at their respective binding sites, then what happens is that because of the ATPase activity of this protein molecule, the ATP molecule is broken down into ADP, a phosphate bond is broken down and energy is released. And by the consumption of that energy, these 3 sodium ions, they move to the outside and 2 potassium ions, they move inside, right? So this is the function of this sodium potassium pump, and it has 2 subunits, as you can see. So the smaller subunit is basically anchoring the whole structure into the membrane, and this is the larger subunit which is basically responsible for transporting the ions inside or outside. And another, factor about, or the point about this pump is that it is electrogenic in nature. By that, we mean that it is pumping 3 positively charged ions outside and 2 ions inside. So can you tell me what is the net effect? The net effect is that the cell is losing positive charges or cell is gaining positive charges? Yes, PDS, I can repeat my question. Since the pump is, transporting 3 positively charged sodium ions out of the cell and 2 positively charged potassium ions inside the cell. So, what is the net effect? Is the cell losing positive charges or it is gaining positive charges?
Ma'am, it may lose positive charge.
Losing
Yes,
positive.
ma'am, they are losing positive charges, so that the inside will become negative.
Yes.
And outside
Yes,
is more
that's right.
positive.
Yes, that's right. There is net loss of positivity from inside. So the inside will become more negative. So that is why we call this pump as electrogenic in nature, because it is creating a negativity inside. It is causing a polarity across the membrane. So that is why we call this pump as electrogenic in nature. Now another point is that this pump maintains cell volume. How is that possible? It maintains cell volume by pumping these 3 sodium ions out of the cell, because if this pump was not there and it was not pumping these ions outside the cell, so these ions will keep on accumulating inside the cell, and it will also drag some water along with it, because when there is increased accumulation of ions inside the cell, then the water follows the sodium ions, and then that causes the cell to swell up and rupture ultimately. So you can see that this pump is also very important to maintain the cell volume, because it keeps pumping these sodium ions out of the cell so that these ions, they do not, accumulate inside the cell. They do not attract water molecule, and the cell actually maintains its volume. So that is the benefit of this pump, and you should be very clear about the activity of this pump, because this pump is is present almost in every cell of the body, and it will be mentioned again and again in every unit of physiology. So you should be very clear. इसको बिल्कुल रट्टा लगा लें कि it just pumps 3 sodium out and 2 potassium in, and it has this ATPase activity, and it utilizes energy to causes this active transport. And we call this as active transport because it is directly consuming the energy. Now we will also see the other type, which is the secondary active transport. So we will see that what happens in that case. So apart from this sodium potassium pump, there are other examples of primary active transport. The first one is the calcium pump, and the other one is the hydrogen pump. So calcium pump is not only present in the cell membrane, but it is also present in the membranes of mitochondria and sarcoplasmic reticulum. Sarcoplasmic reticulum is basically the endoplasmic reticulum which is present in the muscle cells. So what it does is that it pumps the calcium out of the cytosol or out of the cytoplasm, either outside the cell or inside the sarcoplasmic reticulum and mitochondria. So this is basically, this pump is decreasing the concentration of calcium ions in the cytoplasm. It is pumping the calcium ion into the ECF and inside these organelles, right? तो जब organelles के अंदर चला जाएगा calcium, cell से बाहर चला जाएगा तो cytoplasm में या intracellular fluid में उसकी concentration कम हो जाएगी, ठीक है? Or like sodium potassium pump, its carrier protein has ATPase activity. And there are also hydrogen pumps in the body. They are present in the gastric glands of the stomach. Obviously, this is where the HCl is synthesized, and the intercalated cells of the distal tubule and cortical collecting tubule. These are the tubules of nephron present in the kidney, and over there these intercalated cells where this hydrogen pump is present, and it is basically pumping these hydrogen ions out of the body, like into the filtrate or into the tubule, so that the body can get rid of excess hydrogen ions from the body. So these are the 3 examples of primary active transport: sodium potassium pump, calcium pump, and hydrogen pump. And all these 3 pumps are directly using ATP to pump their respective ions against the concentration gradients. So this is it. And now moving on to secondary active transport. It is basically the type of active transport in which the energy that is utilized for the transport of a substance, it is derived secondarily from the energy that has been stored in the form of concentration difference of some other ion. Now, it is very difficult to comprehend this secondary active transport. By studying their examples, it would be easier for you to understand, okay? So, if the secondary active transport has 2 types, co-transport and counter-transport. I will just explain the fact that how it is using energy indirectly. Just, wait for a few minutes. So what happens is that this is the carrier protein, and it has 2 binding sites. On one binding site, the sodium ions attach, and on the other binding site, the glucose attaches. So I can just repeat it for you. That's all I can do. So we are studying about the secondary active transport, which has 2 types, co-transport and counter-transport. Let me just show you. So the secondary active transport has these 2 types, co-transport and counter-transport. So I have jumped to these types directly because I would like to explain with the help of these examples that how it is using energy indirectly. So So the glucose attaches on one side, sodium attaches on one side, glucose attaches on the other binding site, and then there is a conformational change, and both these molecules, they move inside the cell. Now the point is that the sodium is moving along the concentration gradient, and it is also dragging the glucose along with it. So basically, both the molecules which are being transported, so their direction of transport is the same, right? So that's why we call it as co-transport. And you see that the sodium is moving along the concentration gradient, but that gradient was basically developed by the sodium-potassium pump. So basically, this gradient was established by the utilization of energy. It was not, established just like that, right? So, since this gradient was established by the activity of sodium potassium pump, energy was utilized. Now over here in secondary active transport, this sodium is moving because of that energy stored in the form of concentration gradient, okay? And it is also dragging along with it the glucose molecule as well. And both of them, they are moving in the same direction. So that's why we call it as co-transport. So I hope you are getting my point when I'm saying that in secondary active transport, the ATP is used indirectly. Is that point clear to you?
Yes, ma'am. Kind of.
Now let me, show you the other example, and this is the example of counter transport. You see, so in this case, the two ions, they are moving in opposite direction. One ion is coming in and the other one is going out. So this is counter transport because the two ions are moving in opposite direction. And in this, what is happening? The sodium ions are moving inside along its concentration gradient, whereas because of this movement and the conformational change that is taking place in this carrier protein, the calcium is going out of the cell against the concentration gradient. And so you see the energy, where the energy is coming from. The energy is coming from that concentration gradient, and that concentration gradient was established by the activity of sodium potassium pump. तो यू सी एनर्जी तो कहीं ना कहीं पर यूटिलाइज हुई थी, ये तो नहीं है कि एनर्जी यूटिलाइज नहीं हुई. So initially, by the use of energy, this gradient was developed, and now it is this gradient of sodium which is causing the movement of these two ions in the opposite direction. So that is why we say that energy is utilized indirectly in secondary active transport. Now over here you can see again that over here the sodium is attaching, inside the hydrogen ions are attaching, so the sodium ions are moving inside along the concentration gradient, and meanwhile at the same time, because of the conformational change in this carrier protein, the hydrogen ions are moving out of the cell. So in this way, this is another example of secondary active transport, and specifically this one is the counter transport because the two ions are moving in opposite direction. So this is the reason we say that in this particular example, the energy is indirectly utilized. Now I will go back to those slides which are basically explaining this mechanism. So if you again try to read it yourself, if you cannot hear me properly, energy is derived secondarily from energy that has been stored in the form of concentration difference of a secondary ion or molecule, right? So this is the type of active transport in which energy is utilized indirectly, not directly like that, like it happens in primary active transport. So again, it has two types, co-transport and counter-transport. So co-transport में जो है diffusion energy of one ion can pull other substance along with it across the cell membrane. So examples of co-transport are sodium glucose co-transport and sodium amino acid co-transport, as you can see over here. So this is sodium glucose co-transport. And there are some other carrier proteins on which sodium and amino acids attach. So that is the example of sodium amino acid co-transport. And these, transporters, they are found in epithelial cells of intestinal tract and epithelial cells of the renal tubules, where this co-transport is taking place. Okay?
Ma'am,
जी बेटा,
I have a question.
जी पूछिए.
मैम अब ग्लूकोज है और कैल्शियम है और हाइड्रोजन है, लाइक ये लोग क्यों लाइक आउट ऑफ द सेल और इनसाइड द सेल आते जाते, लाइक इज इट बिकॉज ऑफ कंसंट्रेशन ग्रेडियंट या फिर ऐसी इनकी जरूरत होती है और सोडियम इनकी हेल्प करता है?
मुझे ज़रूरत तो होती है ना ग्लूकोज की सेल के अंदर, क्योंकि ग्लूकोज जब सेल के अंदर जाएगा तो वो उसका एनर्जी प्रोड्यूस करने के लिए यूटिलाइज होगा. जैसे ऑक्सीडेटिव फॉस्फोराइलेशन हो रही होती है माइटोकॉन्ड्रिया में, तो वहाँ पे ग्लूकोज ब्रेक डाउन होता है और उससे एनर्जी रिलीज होती है, एटीपी बनता है जो सेल की वेरियस एक्टिविटीज के लिए यूज होता है. So it has to enter the cell, and sodium is, you can say, it is helping the glucose to enter the cell. And I have also mentioned it before कि जो ग्लूकोज है वो फैसिलिटेटेड डिफ्यूजन से भी सेल के अंदर एंटर होता है और ये को ट्रांसपोर्ट से भी एंटर होता है. सो बोथ mechanisms are utilized to transport glucose inside the cell. I hope I have answered your question.
Yes ma'am, I got it.
देखो अमाइनो एसिड्स भी न्यूट्रिएंट्स हैं, ग्लूकोज भी न्यूट्रिएंट्स हैं, तो दे नीड टू एंटर द सेल, तभी तो सेल उसको यूटिलाइज करेगा एनर्जी प्रोडक्शन के लिए. अमाइनो एसिड्स से जो है वो मतलब दे विल बी यूटिलाइज्ड फॉर synthesis of various proteins, so they need to enter the cell so that they can be utilized by the cell. ठीक है. और सिमिलरली जब हम काउंटर ट्रांसपोर्ट की भी बात कर रहे होते हैं तो देखें सोडियम कैल्शियम काउंटर ट्रांसपोर्ट है. तो नॉर्मली आई टोल्ड यू कि जो कैल्शियम की कॉन्सेंट्रेशन है ECF में ज्यादा होती है एज कम्पेयर टू ICF. So it is basically this pump which is working to pump the calcium out of the cell. ये secondary active transport भी अपना रोल प्ले करती है और हमारे पास कैल्शियम पंप्स भी होते हैं, वो भी अपना रोल प्ले करते हैं और कैल्शियम को जो है सेल से बाहर पंप करते हैं. Similarly, we have these counter transport mechanisms in which the sodium is pumped inside the cell and hydrogen is being pumped outside the cell. Because if the pH of the cell and the body fluids needs to be maintained, तो उसमें जो है हाइड्रोजन को निकालना पड़ता है सेल से बाहर. तो मतलब for these various purposes these transporters are important. ठीक है. So when we are talking about the active transport, it can be divided into two types: primary active transport and secondary active transport. Primary active transport directly uses ATP and moves the particles against the concentration gradients or electrical gradient or pressure gradient, whatever. And their examples are sodium potassium pump, calcium pump, and hydrogen pump. Then the other type is secondary active transport, which indirectly uses the energy which has been stored in the form of concentration gradient. So now that gradient is then utilized to cause the movement of another ion and or particle across the membrane. So this type of movement is known as secondary active transport, and it has further two types: co-transport and counter-transport. So in co-transport, the particle which is moving along the concentration gradient and the other particle which is just moving along with it, both of them are moving in the same direction. And in counter transport, one ion which is moving along the concentration gradient and the other ion which is moving along with it, they are moving in the opposite directions. So that is why we call that as counter transport. So this was all about active transport. If there is any question, you can ask. I know it's not important कि अभी जो आपने जितना भी मैंने आपको पढ़ाया है वो सारा कुछ आपको एकदम से सारा बिल्कुल समझ आ जाए और क्रिस्टल क्लियर हो सकता है बहुत सी चीजें नहीं भी क्लियर हो, लेकिन आप ऐसा कीजिएगा कि आपको खुद पढ़ें बुक से, और कोई भी ऐसी चीजें जो आपको नहीं समझ में आती तो कल आई विल ट्राई के कुछ मैं लेक्चर्स को आगे पीछे करके आपका लेक्चर लेने की कोशिश करूंगी, बिकॉज आई एम ऑल्सो टेकिंग द लेक्चर्स ऑफ फर्स्ट ईयर MBBS एट द सेम टाइम. तो आई विल ट्राई टू मैनेज समथिंग, कोई टाइम आगे पीछे करके आप लोगों की क्लास लूंगी, और अगर कोई क्वेश्चन हुआ, कोई चीज़ आपको समझ में नहीं आई या आप चाहते हैं कि मैं रिपीट कर दूं, तो आई विल डेफिनेटली डू दैट। तो आप मतलब उसके लिए ज़रूरी है कि अगर आप खुद पढ़ेंगे तो आपको पता चलेगा कि क्या क्वेश्चन पूछना है ना। तो
Yes, ma'am.
this is just one last slide. I would just like to go through this so that this chapter is almost finished after this. Now what happens is that there are certain areas in the body in which the cells are present in sheets. So the mode of transport over there is we need to see that how the movement of substances is taking place when we are talking about layer of cells or sheets of cells, right? So you can see over here in this diagram, there are 1, 2, 3, 4 cells which are lined up, right? And this is the luminal surface of the cell. Lumen, which is facing the lumen. Like let's suppose if we are talking about intestines, so this is the side of the cell which is facing the lumen of the intestine. Okay, or if we are talking about renal tubules or any kind of hollow organ, so this is the side which is facing the cavity or the lumen of that particular organ. Now these two sides are known as the basolateral side. These are the lateral sides of the cell, and this is the basal side which has a basement membrane behind it to provide support to it. So when we are talking about the movement of particles or ions in these layers of cells, so how that is taking place? The first step is the active transport of ions. Right now we are just talking about the sodium ions, although there are so many other ions as well. But just to make things easier and easy to understand, they are just talking about the sodium ions. So at the basolateral sides, the active transport of sodium ions is taking place, right? So when the sodium ions are moving into these intercellular spaces, the water molecules, they move along with it because of the process of osmosis, right? So this osmosis and active transport is taking place on the lateral sides and on the basal sides. You see active transport and osmosis, so that is causing the movement of sodium and water out of these cells. And then what happens because of the gradient which is developing because of this active transport, the sodium ions from the lumen, that basically diffuses into the cell because of that concentration difference. So it causes the net diffusion of sodium ions in this direction, and from inside the cell it is transported by active transport into the intercellular spaces and into the basal side so that it can move out of here and enter the blood or whatever its destination is. So if you see, there is the net movement, that the direction of the movement of ions or the sodium is from the lumen to the outside of the cell where the blood vessel might be present. It can enter the blood. So this is how it works basically. So there is active transport and osmosis taking place at the basolateral surfaces, and passive transport of sodium taking place at the luminal surface. So this is what happens when we are talking about the layer of cells which are present in various spaces and various areas of the body, like intestinal epithelium, epithelium of the renal tubules, epithelium of the exocrine glands, because they also, release their secretions out of the cells, and membrane of the choroid plexus, which is a plexus or network of capillaries present in the brain. And the steps are these two steps, which I already told you: active transport on one side of the cellular sheet and diffusion, which can be simple or facilitated on the other side of the cellular sheet. The other side is basically the luminal side. So this is how the movement of ions or substances takes place through the cellular G. Is there any question? जी जी बेटा पूछिए। अच्छा प्लीज मुझे जल्दी से बात बताइए कि डू यू वांट मी टू शेयर द लिंक वंस अगेन और यू आर डन विद दिस? कल अब करें क्लास?
हम कल
करें ना, थक गए हो आप लोग? चलें ठीक है, बाकी कल कर लेंगे सो दैट आई डोंट हैव टू शेयर द लिंक अगेन। ओके
यस मैम।
ओके,
Okay.
I will try to meet tomorrow, so if there are any questions तो आप कल पूछ सकते हैं, ठीक है? अल्लाह हाफिज एवरीवन।
अल्लाह हाफिज मैम।
अल्लाह हाफिज।